Did you know your body fights microscopic invaders every second of the day? Imagine this. Your immune system is like a highly trained SWAT team, constantly scanning for enemies. And how does it know who's a friend and who's a foe? It's all because of something called an antigen, the ultimate most wanted poster in your body's defense playbook. But wait, some antigens can sneak past, while others trigger alarms so powerful they unleash an all-out immune war. And here's the craziest part. Scientists have figured out how to trick your immune system using tiny molecules called Haptens and Adjuvants, turning your body into an unstoppable defense machine. Ready to discover how it all works? Let's dive in. An antigen is any foreign substance that can trigger an immune response in the body. Antigens are like secret codes on invading bacteria, viruses, or toxins that alert the immune system. Once detected, your body creates antibodies, special proteins that fight and neutralize these invaders. Antigens can be proteins, sugars, polysaccharides, lipids, or even nucleic acids. The most important thing about antigens is that they need to be recognized by our immune cells, mainly the B cells and T cells, to start the defense process. Antigens have two key properties. The first is antigenicity, which is their ability to bind specifically with antibodies or immune cells. Think of this as a key fitting into a lock. It's how the immune system identifies the intruder. However, just binding isn't always enough. That's where immunogenicity comes in. This is the ability of an antigen to not only bind, but also to provoke a full immune response. While all immunogens are antigens, not all antigens are immunogens. Vaccines are great examples of immunogens because they trigger the immune system to react and build protection without causing the actual disease. Now, what exactly are immunogens? Immunogens are antigens that can actually provoke an immune response. They activate B cells to produce antibodies and T cells to attack infected cells. For example, the COVID-19 vaccine uses a spike protein as an immunogen. It teaches your immune system to recognize and attack the real virus if it ever enters your body. But sometimes, the immune system needs a little help. That's where adjuvants come in. Adjuvants are substances added to vaccines or immunogens to boost the immune system's reaction. On their own, they don't cause an immune response, but they help the immunogen work better. A common example is aluminum salts, which are used in many vaccines to increase their effectiveness. Adjuvants make vaccines stronger, reduce the number of doses needed, and provide longer-lasting immunity. Now let's talk about haptens. These are small molecules that by themselves can't trigger an immune response. But when they attach to a larger carrier molecule, they suddenly become immunogenic. A common example is penicillin. It acts as a hapten when it binds to proteins in the body, which can sometimes lead to allergic reactions. This concept helps explain how drug allergies and certain autoimmune diseases develop. There are several factors that influence how strong an immune response is, also known as immunogenicity. First is foreignness. The more different something is from our own body, the stronger the reaction. Second is the size. Larger molecules are usually better immunogens. Third is chemical complexity. More complex molecules create a better response. Fourth is degradability. The antigen must be broken down by antigen-presenting cells to be shown to T cells. Other factors include the host's genetics, the route of administration, like injection or inhalation, and whether an adjuvant is used. Next, let's explore epitopes, also known as antigenic determinants. Epitopes are the specific parts of an antigen that immune cells recognize. Imagine an epitope as the exact shape or piece of the puzzle that the antibody or T cell receptor fits perfectly. A single antigen can have many epitopes, which means different immune cells can recognize different parts of the same invader. For example, a virus might have multiple spike proteins, each with its own unique epitopes, allowing the immune system to target it in different ways. Now, focusing on B cell epitopes, these are generally found on the outer surface of the antigen, where antibodies can easily bind to them. They are often hydrophilic, meaning they are attracted to water and are accessible. B cells recognize these epitopes in their natural, three-dimensional shape. An example is B cells binding directly to the protein coat of an influenza virus. On the other hand, T cell epitopes are different. They are linear peptide fragments made up of 8 to 20 amino acids. These peptides are processed and presented by MHC molecules on the surface of antigen-presenting cells, APCs. T cells don't recognize the whole antigen but the processed pieces. For example, when a virus infects a cell, fragments of the virus are displayed on MHC molecules to activate T helper cells or cytotoxic T cells. So, these microscopic players are the masterminds of your immune system's defense. From fighting off deadly viruses to creating life-saving vaccines, understanding these concepts helps us appreciate how our bodies protect us and how science can make it even stronger.